Welcome to our Focus20 webinar episode on digital analysis in medical technology, hosted by Magdalena Berg, Managing Director of Adalogic, and her colleague Michael Radke.
Welcome and hello to today's webinar on digital layer analysis in medical technology. This is a webinar in our Focus20 series geared at delivering you valuable insights at a very compacted time. Just 20 minutes fit in every busy schedule. Today's webinar will be recorded. Your participation is completely anonymous. We will be sharing this recording after the webinar so you can review it or in turn share it with your colleagues who were not able to attend. There is a roundabout 5-minute Q&A session at the end of this webinar. You can post your questions at any time via the Q&A box. Yes, that's it from my side. Without further ado, Michael, the floor is yours.
Yes, hello and welcome also from my side to our webinar concerning medical layer analysis. The main topic is, as said, the digital layer coating analysis for implants. One main goal is to show how precise layer analysis is indispensable today and how OptiMa can be used as a tool of reproducible, fully documented measurements. Why layer analysis matters? Coatings determine the biocompatibility, the wear behavior, and as well the osseointegration. Small deviations can have a big impact in the result. If it's too smooth, the bone has difficulty growing in. The wrong porosity can cause an unstable fixation, and also the wrong layer thickness. You can have more friction and wear as you want to have. The regularity background from MDR and FDA requires a standardized, traceable, and reproducible, repeatable measurement process, which is very, very important for your audits you may have with customers or with the FDA. What are the challenges of manual methods? You have a strong operator dependence when you have different lab people working on the examination. The porosity assessment often is not consistent. The documentation is very high time consuming and not really audit proof. The media breaks between microscope evaluation and report can cause failures as well. And you have a risk in regulated environment, just a lack of traceability, which is very important for medical equipment. Which opportunities do we have through automation? OptiMa automates layer analysis according to standards. It's the ISO 1463 for the layer thickness in cross-section. The ISO 26423, the calotte grinding for thin hard coatings. And the ASTM F1854, which is including the porosity and tissue interface height. The benefits with OptiMa are reproducible results, objective instead of subjective, and an audit-ready documentation. And by the way, it's up to 80% faster. Here you see a practical example of medical technology, just in the easy way, because we are not experts in that case. You see a porous titanium coating, which is used for hip or knee implants. Here the critical factors are that you need an open pore fraction, a distribution of pore regions, a layer structure, and a layer thickness. If it's too smooth, no bone ingrowth. If the porosity is too high, you have a loss of mechanical stability. And in these cases, OptiMa provides automatic segmentation of pore regions, the porosity in percent, the layer thickness, and the tissue interface height. Precise coating thickness measurement on implants. There we have in general thin hard coatings with a size of 2 to approximately 10 microns. For that examination, the cross-section often is not suitable because the preparation of the sample is very critical in that thin amounts. Here, the calotte grinding is the right choice and the most precise method. OptiMa calculates the thickness automatically according to the ISO 26423. And especially important for joint surfaces, every single micron influences the friction wear and the service life of the implant. Here you see a measurement of the pore size and the pore volume. This sample is prepared in the cross section of a porous titanium surface. OptiMa here provides and automatically detects the pore regions, determines the porosity, measures the layer thickness, and calculates the tissue interface height. The result is a clear statement on osseointegration, and it has a direct relevance for MDR and FDA documentation. What are the key advantages of OptiMa? You have up to 80% time saving. You have a high reproducibility and repeatability thanks to reduced operator influence. You get audit-ready reports, and it's reliable basis for quality assurance and regulatory approval in that case. So now we can switch over to a live demo and can measure some pictures live here. Now we switch to the OptiMa software. Here you can choose the module. Here you see layer analyzes cross section, the layer porosity, layer analyzes calotte grinding, and for today not relevant, but also including in the software the cast aluminum analysis and also the homogeneity analysis. We start with a calotte grinding measurement. Here we can choose some pictures. We choose it and we choose all in one step. You also can choose more or less pictures. Now it's included in the software. Here we see a picture with a quite high quality. We can measure it automatically, and within seconds we get a result of the thickness of the layers. We can save it as well to our series. Here's the next picture. We also can measure it automatically, get here a very precise result of the layer thickness. Just the third one. Also within seconds you get the result here. And I know from my experience that the work in the lab and it's not always black or white. You get also pictures which had not that high quality related to the picture or the preparation of the sample. Here it's too difficult to detect the result in an automatic mode. There we can switch over to an interactive mode. Here we can place 3 dots with a click, and then the layer is measured. Also with the second layer, we can do it in the same way. And also here, in a manual way, we get within a few seconds a result of the layer thickness. Here we also switch into the interactive mode. And measure it, and also with the other shift. And then the measurement is ready. And now we measured 5 pictures in a very, very short time. And we also can, as I told in the presentation, can generate an Excel protocol which is now ready. Here we have one page with all measurements, and then we have for the single pictures one lab report with a measured picture, including the results, which you can save for your documentation, or also from single measurements you can create a PDF, for example, to send it as a certificate to your customer. I think that's it from the calotte grinding. Now just let's have a short look into the porosity module. You can choose it here. Then again, we can add one picture. Then we include the tissue interface height with one click. And now we can analyze the current image, and as well here within seconds, we get the layer measurement, the minimum, maximum, the mean, the standard deviation, the porosity, which is a very important factor for layers. There we have 44%. Also, we have a mean interception of the length of the pores, and the tissue interface height is in that example 595, which is similar to the maximum layer thickness. Now, that was it from the live demonstration. Now I can give the microphone back to Magdalena.
Thank you very much. So I'm trying to structure your questions a little bit so as not to have them all over the place. Let's start with the first one, which is, can OptiMa be integrated into existing laboratory or production processes?
Yes, that can be done very easy, because OptiMa is independent from various hardware, microscopes or computers. It also can be integrated or can transfer the data to QMS and PLM systems which are in use. So the implementation is possible without major process changes in general.
Perfect. Second question, what prerequisites do we need in our laboratory to use OptiMa?
What we need for OptiMa is a digital image on an SEM light microscope or any camera source, and OptiMa runs on standard PC hardware. There are no special devices required. So existing microscope can continue to be used without modification. And one big advantage of OptiMa is that OptiMa also doesn't care which kind of microscope it is, from which manufacturer. We don't care.
Next question. Let's take this one. How do you handle updates or future changes in standards?
Yeah. OptiMa is continuously developed and receives regular updates, so approximately 1 to 2 per year. And if some norms have some changes or new specifications inside, we get the information right on time so that we can implement it into the new release, and that's always before the norm is working for the industry. And also we include new features in these updates, which you get regularly once a year, automatically sent by us.
Yeah, perfect. Looking at the clock, let's do one last question. The remainder of the questions we will answer via email once we're sending you the recording of this webinar. So last question for today is, how much training is required until our staff can operate OptiMa confidently?
So OptiMa is designed for users to become productive after a short introduction. From our experiences, first introduction is maximum 1 hour, and then you can start working productive with our systems. And if it's a special need for special demands, we can put another training on site on your demands. So after a short time, you get a quite good routine with examinations you have to do.
Perfect. Thank you. Thank you, Michael, for your time. Thank you,
Yes.
everyone else as well, for listening and joining in. Have a great rest of the day. Goodbye, and until next time. Bye-bye.
Okay, thank you. Bye-bye.